Next, we move on to specification 2, which requires us to describe how antibiotics are used to treat bacterial infections. So, if we define an antibiotic, we can say that antibiotics are drugs that alter the body's chemical processes by killing bacteria or preventing them from multiplying. They are produced by certain bacteria or fungi and are effective only against bacterial infections, not viral ones. Antibiotics work by targeting structures or processes found specifically in bacterial cells, which is why they usually do not damage human body cells. Antibiotics are a type of drug, which means they are substances that alter or influence the chemical reactions within the body. They are chemical compounds that are naturally produced by certain bacteria and fungi. Antibiotics act by attacking bacterial cells in a variety of ways. Some damage the bacterial cell wall or cell membrane, leading to the death of the bacteria. Others interfere with essential internal processes, such as protein synthesis or DNA replication. Certain antibiotics stop bacteria from reproducing, which prevents the infection from spreading further. It is important to note that antibiotics are effective only against bacterial infections and do not work against viral infections such as the common cold or flu. Antibiotics are able to work selectively because they target structures and processes that are unique to bacterial, prokaryotic cells, including cell walls and ribosomes. Since human, animal, cells are structurally different, antibiotics usually do not harm the body's own cells. Overall, antibiotics are vital medicines used to treat a wide range of bacterial infections, including pneumonia, strep throat, and various skin infections. Next, we will look at how antibiotics work. Antibiotics function by targeting features that are found only in bacterial cells. Some antibiotics damage the bacterial cell wall or prevent it from forming properly. As a result, the weakened cell wall cannot withstand internal pressure, causing the bacterium to burst and die. Other antibiotics interfere with the bacterial cell membrane. This disruption makes the membrane leaky, allowing essential substances to escape, which eventually leads to the death of the bacterial cell. Certain antibiotics work inside the bacterial cell by blocking protein synthesis. Without the ability to produce essential proteins, the bacteria cannot survive. Some antibiotics also stop DNA replication or interfere with DNA repair, which prevents the bacteria from growing or dividing. In addition, there are antibiotics that prevent bacteria from reproducing, slowing down the spread of infection within the body. By attacking these vital processes, antibiotics either kill bacteria directly or stop them from multiplying, giving the immune system time to remove the remaining bacteria. Human cells are not harmed because antibiotics target structures and processes that are unique to bacteria. This selective action makes antibiotics safe and effective for treating bacterial infections. As you can see in the diagram, a normal bacterium that has an intact cell wall and is functioning properly. When the antibiotic is introduced, it attacks the bacterium by targeting its cell wall. This attack damages the structure of the cell wall, making it weak and unstable. Because the cell wall can no longer support the internal pressure of the cell, the bacterium bursts. As a result, the bacterial cell is destroyed and dies. This shows how antibiotics can kill bacteria by damaging essential structures that are necessary for their survival. Similarly, let us now look at penicillin. Penicillin is a type of antibiotic used to treat bacterial infections. It specifically targets bacterial cells and usually does not harm human cells because human cells do not have cell walls. Bacterial cells, on the other hand, possess a rigid cell wall made of a substance called peptidoglycan. This cell wall gives the bacteria its shape and protects it from bursting due to internal pressure. Penicillin works by interfering with the formation of the bacterial cell wall. It blocks the enzymes that bacteria require to build and repair their cell walls. When these enzymes are blocked, the bacteria are unable to produce a strong and complete cell wall. As the bacterium continues to grow and divide, its cell wall remains weak and incomplete. This loss of structural strength makes the wall unable to withstand the pressure inside the cell. Eventually, the weakened wall ruptures, causing the bacterial cell to burst, a process known as lysis, leading to the death of the bacterium. By destroying bacteria in this manner, penicillin helps the immune system control the infection and prevents the bacteria from spreading further. As you can see here in the diagram, penicillin acts directly on the bacterium by blocking the enzymes that are needed to build the bacterial cell wall. Because these enzymes are blocked, the bacteria are unable to form a strong and complete cell wall. As the bacterium continues to grow, the weak cell wall cannot handle the internal pressure. This causes the cell wall to rupture, and as a result, the bacterial cell bursts and dies. This diagram clearly shows how penicillin kills bacteria by preventing proper cell wall formation.